Hello, in this video we will understand about the rotavirus infection. So this is the virus contain a spike protein VP4 and VP7. And this virus is a double-stranded RNA virus. This is the family of virus we will discuss now. So basically it causes a diarrheal disease which that will lead to problem the digestion and the dehydration of the body due to the electrolyte imbalances. Among infant and young children, nearly every child in the world, at least once of the age of 5. 9 species of the genus differed to as A, B, C, D, F, G, H, and J. Rotavirus A is common, causes more than 90% of rotavirus infection in human. Let's learn about their sign and symptom as well as their infection site. So remember the fecal to oral route contaminated food. The contaminated food mixed with the rotavirus will be ingest in the form of bolus, and the acidity will not tolerate it and it will not kill. And that is why the small intestine will pass into the small intestine. The 2 days incubation period will lead to progression of the infection in the duodenum. The gastroenteritis will be occur. It is the stomach flu. This is the 37 death per year can be possible. Through research shows that sign and symptom is the nausea, vomiting, and the diarrhea as well as dehydration due to the continuously diarrhea, and the fever can be caused due to the progression of the rotaviral disease. The asymptomatic can be include when nausea will lead to asymptomatic, while lead to progress after the vomiting to the diarrhea and dehydration is the symptomatic and non-pyrogenic, while or mild pyrogenic will lead to severe immune response, will lead to pyrogenesis, cause the fever, the high fever. High fever is due to the pyrexia. The pyrogens will produce is known as the pyrogenesis. Pyrogenesis is produced due to the infection, and the infected cell will produce that for increase the temperature. So let's begin to understand the enterocyte, the brush border cell. The rotavirus entry will lead to double layered particles formation in the enterocyte. After this go into the Golgi bodies, the rotaviral produces non-structural protein NSP4. The non-structural protein 4. This non-structural protein 4 will produce and it will target to the another tissue we will discuss also. But remember, this will be damage the enterocyte. The cell lysis will occur and the tight junction will be loose due to the infection and the lysis and apoptosis of the cell. And in this way the calcium regulation will be imbalanced and the calcium will move out due to the NSP protein 4. While when the exocytosis of the rotavirus will produce the different mechanism will lead to produce NSP4 protein, will trigger the phospholipase C enzyme to activate to the Golgi bodies to efflux of the calcium more. On the other hand, here is the tight junction will be damaged. In this way, the permeability of the enterocyte will be increased, will lead to water imbalance problem, and the enterocyte lysis and malabsorption of the nutrients will be defecate. And in this way, the malnutrition will occur. And in this way, the NSP4 protein basically produced further, and in this way the dehydration due to the outflux of the water and chloride ion through NSP4 protein will lead to imbalance the electrolyte. The electrolyte imbalances will lead to also inflammation and damage the enterocyte. When damage the enterocytes mean the permeability will increase, will lead to further pathogenesis can be caused. For example, bacterial pathogenesis like Salmonella bacteria entry. Other secretory and osmotic diarrhea can occur. Also here you can see this is the neurons which that peptides and amines produce, and in this way the neurotransmitter will lead to calcium move out, and in this way the peptide and amines will be trigger will lead to enteric nervous system problem. On the other hand, remember this is the immune response also occur due to the VP4 and VD4 and 7 detection through toll-like receptor through dendritic cell to activate the CD4 T lymphocyte, to activate the B cell through CD4, to produce B memory cell and plasma cell, to produce the IgM, A and G antibodies for the protection of the rotavirus. Remember, the diagnosis is the stool test first and by enzyme immunoassay, while on the other hand the detect all serotype of rota A. On the other hand, the electron microscopy can be identify the rotavirus, and the PCR as well as real-time PCR detection for the identification of the infection and justification and the treatment, the management of dehydration due to the electrolyte transfusion through venous transfusion and the oral dehydration therapy, salt and sugar drinking, oral dehydration solution and zinc using, and electrolyte and blood sugar monitoring, while the vaccination before infection is a prevention process which can be prevented due to the rotaviral infection prevention vaccine for produce the immunoglobulin against for the suddenly attack of the rotavirus to combat due to the immunoglobulin will be produced already due to the immune system induced vaccination. Thanks for watching. Please make sure to subscribe, like and share. Bye.